Hello friends, in the previous video we learned about fertilization. Now we are going to learn further embryonic developments. Embryogenesis is quite longer and complex than other concepts. Here we will learn the process in 4 steps. These are cleavage, morulation, blastulation and gastrulation. We will not take a lot of time and just gonna learn in shortcut and cover through important and direct points. So let's go through first stage, that is cleavage. The development of fertilized egg starts with cleavage. It is a kind of mitosis cell division, but my dear friends, it is different from general mitosis as cleavage occurs in all the cells, but mitosis doesn't. The interphases are comparatively shorter and do not involve growth. The cell division and DNA replication are rapid comparatively, and oxygen is highly consumed in cleavage. So now let's go little deeper to understand it. First, the zygote vertically divides, that is meridionally, to form first blastomer. Here the blastomers simply mean bubble or grape-like structure where each grape is known as blastomer. In second cleavage, blastomer meridionally divide to form second cleavage. This again horizontally divide to form micromer and macromers. Again by vertical division, 16 micromers and 16 macromers are formed. In fifth step, furrowing happens. This furrowing is directly connected to morulation stage. Now in morulation, it is simply the solidation of cleavage zygote like in shape of mulberry fruit, or we can understand that it is just a result of cleavage stage. Now here white yolk laden is vegetal pole of morula and black yolk is animal pole of morula. You can clearly note vegetal and animal pole in the figure. I hope you understand it. And here is the main point to be noted, that morula can be differentiated from blastula just by the absence of cavity, as shown in figure. After the formation of morula, blastula is necessary to be formed for further embryonic development. So, blastulation is the loss of cleavage structure in order to form single blastula. In this phase, irregular development of micromers and megamers happens. This tends to create random cavity inside it, which is called blastocoel. In case of frog, this cavity is known as celloblastula. When blastula is formed, the blastocoel consisting zygote is known by blastocyst, and the blastocyst in short is defined by hollow cellular mass. Here is the point to be noted that fate mapping is done one and only in blastula stage. Dear friends, fate mapping is a method used in developmental biology to study the embryonic origin of various adult tissues and structures. In this fate mapping, the color helps for predetermination. At first, melanin pigment consisting layers are classified as ectoderm, gray crescent representing notochord are classified as mesoderm, and yolk-laden megamers or dark gray crescents are classified as endoderms. You can watch this simple animation and can understand in more depth. Just now you saw how blastula converted into gastrula stage. Now we will learn exactly about this in details. Let's understand first that what is gastrulation. So my dear friends, gastrulation is simply transformation of blastula into multi-dimensional structure and diploblastic gastrula, and therefore establishes 3 germ layers. This can be clearly understood from that animation. So this cycle complete in about 30 hours. In this time, different movements that we call oviposition happens. These are like at first epiboly or overgrowth, at second emboli or invagiation or entachy, and third one rotation of gastrula. Let's learn these things in details. In the epiboly, micromer divide faster to cover vegetal pole except yolk plug region rather than slow developing macromers. In the emboli, crowd mesodermal cells grows inward, forming groups called archenteron, which opens in blastophore bounded by dorsal and ventral lip. The pressure pulses macromers as yolk plug in yolk plug stage. Involution is that process when endomesodermal cells rolls over the leaves. It clearly develops germ cells. To understand it, you can look at the animation clip once again. In the rotation of gastrula, or we called it as deuterostomes, is simply the 90-degree rotation of archenteron cavity. After this process, ectoderms formed arises to exoskeleton, mesoderms develops into different organs, and endoderm forms inner lining of organs. The detailed embryogenesis of these germinal cells are provided in a table. Read the table, you can get into depth by yourself. So my friends, this much in this video. We talked a lot about embryogenesis with all necessary notes. This fate mapping is simply the preparation for organogenesis, which we will discuss in next video with next lecture. Thank you for watching. See you again with next video of embryology, that is organogeny.